Hello, YouTube fan. Get ready for an amazing video experience. We're thrilled to present to you our video dedicated to hydroxylation of estradiol. Enjoy. The hydroxylation of estradiol is one of the major routes of metabolism of the estrogen steroid hormone estradiol. It is hydroxylated into the catechol estrogens to hydroxyestradiol and for hydroxyestradiol and into estriol hydroxyestradiol reactions which are catalyzed by cytochrome enzymes predominantly in the liver, but also in various other tissues. Without wasting any more time, let's jump into the fascinating world of 2-hydroxylation. Addition of a hydroxyl group represents the major hepatic pathway for estradiol metabolism, as mediated by CYP, CYP, and CYP. Extrahepatic 2-hydroxylation is chiefly mediated by CYP and CYP. 2-hydroxyestradiol 2O can experience three metabolic fates: methylation to yield 2-MO, oxidation to form quinones, or hydrogenation to yield 2O. 2O can bind to estrogen receptors but with markedly lower affinity. This metabolite has several physiological consequences: the ability to influence intracellular signaling, adenohypophyseal hormone secretion, radical and quinone formation, and inhibition of tumor formation. Weak carcinogenic activity has been shown, likely due to radical formation and induction of single-strand DNA breaks. Inactivation of 2O is catalyzed by catechol methyltransferase COMT, with COMT exhibiting a faster rate for the methylation of 2O versus 4-OHE. COMT, a blood-borne enzyme, mediates the most common form of 2 or 4-hydroxyestradiol inactivation, in addition to glucuronidation and sulfation. However, this inactivation can allow for the accumulation of 4-O, as 2O inhibits 4-O methylation by COMT, but 4-O does not inhibit 2-OHE methylation in return. Antitumor activity of 2-MeO is thought to be mediated by antiproliferative and antimetastatic effects. Inhibition of cellular proliferation and metastasis appears to be via induction of caspase 8, followed by caspase 3 and eventually DNA fragmentation. Induction of apoptosis by 2-MeO may be dependent or independent. 2-MeO has also been found to inhibit aromatase activity, thereby lowering the in situ synthesis of E in cancer tissue. 2-MeO has a higher binding affinity for sex hormone binding globulin SHBG than E and 2-OHE and has no affinity for the estrogen receptor. 2-MeO is also a potent inhibitor of angiogenesis in tumor tissues. Administration of this estradiol metabolite prevents vascular smooth muscle growth. This inhibition of angiogenesis is eliminated by co-administration with cytochrome and COMT inhibitors, thereby confirming the involvement of cytochrome enzymes in the blockade of tumor blood supply. Further antitumor activity of 2-MeO has been identified through immunomodulation. The cytokines IL-6 and TMF, as well the prostaglandin Ch, are capable of stimulating aromatase activity. Since macrophages and lymphocytes are present in breast tissue, this provides a concerning means of upregulating in situ estradiol biosynthesis. 2-MeO appeared to be able to have the basal aromatase activity in mammary fibroblasts, possibly through destabilization of the microtubules that mediate translocation of the cytokine receptors to the plasma membrane. Inhibition of cytokine receptor synthesis and blockade of the autocrine and paracrine actions of cytokines and Ch were also observed. Let's now shift gears and explore 4-hydroxylation through a critical lens, uncovering its strengths and weaknesses. The enzyme most responsible for estradiol 4-hydroxylation is CYP. In humans, CYP and protein exhibit constitutive expression in the lung and kidney, as well as estrogen-regulated tissues such as breast, ovary and uterus. Whereas 4-hydroxylation constitutes the minor pathway in the liver, the greater proportion of CYP expression in extrahepatic tissues shifts the balance in favor of 4-OHE formation. 4-OHE is thought to be the most carcinogenic of all the estradiol metabolites, especially considering that CYP exhibits overexpression in breast cancer tumors. 4-OHE, like 2-OHE, can be physiologically active as well as tumorigenic. 4-OHE is capable of binding ER with a reduced dissociation rate and prolonged activation, thereby inducing cellular growth and proliferation, adenohypophyseal hormone secretion, and prostaglandin production. Das et al. Implicated 4-OHE in the induction of estrogen-responsive genes, a response that exhibited partial or no abrogation by co-administration with an antiestrogen, providing evidence for the ability of 4-OHE to carry out genetic upregulation via a pathway independent of ER signaling. Effects independent of ER binding include breakage of single-stranded DNA, especially when interacting synergistically with nitric oxide in human breast cancer cells, and the production of quinones and free radicals. CYP can be induced by E. After binding to estradiol, interacts with the CYP-ERE to stimulate CYP expression. Thus, although E causes genetic changes conducive to its own inactivation, the decrease in estrogenic activity yields a toxicologically active metabolite that constitutes an additional pathway of estradiol-dependent carcinogenesis. 4-OHE shares the metabolic scheme of 2-OHE, methylation to form methoxyestradiol formio, oxidation to quinones, or hydrogenation to 4-OHE. Conjugation by the ubiquitously present COMT represents the most common extrahepatic pathway of 4-OHE inactivation. However, if estrogen homeostasis is imbalanced by an increase in CYP and a decrease in COMT, a greater degree of genotoxic quinone formation from 4-OHE will occur. 4-O can be oxidized by microsomal CYPs or peroxidase to yield estradiol 30 for semiquinone. This semiquinone can undergo redox cycling with oxygen to form estradiol 30 for quinone E34Q and superoxide. E34Q can be converted back to 4-O in a single step by quinone reductase, or into sequential steps catalyzed by reductase via the semiquinone intermediate. GSHS transferase activity can abrogate E34Q levels via formation of glutathione conjugates. E34Q is a potent nucleophile and will readily react with electrophilic DNA. This yields the formation of the DNA adducts 4-O1-GUA and 4-O1-NADE via a Michael addition. Destabilization of the glycosyl bond between the nitrogenous base and ribose sugar creates apurinic sites as the unstable adducts are lost from DNA. 4-O1-GUA has a relatively slow depurination half-life of approx. 3 hours, allowing enough time for base excision repair mechanisms to correct the change. However, 4-O1-NADE exhibits instantaneous depurination, leading to error-prone repair and the induction of mutations. Indeed, E34Q has been shown to cause A to G mutations in the gene coding for HRAS, RAS being vital to the correct regulation of the cellular response to growth factors. Though 2 and 4-O have similar redox potentials and thus similar redox cycling activity, the greater carcinogenic capacity of 4-O can be attributed to its increased reactivity with DNA. Another harmful effect of estrogen redox cycling is the production of superoxide and hydroxyl radicals. Reductase catalysis produces superoxide radicals, which can, in the presence of superoxide dismutase and Phe, form highly reactive hydroxyl radicals capable of damaging virtually all macromolecules. In the next portion, we'll be immersing ourselves in the realm of hydroxylation and examining its broader implications. Through the action of CYPA, CYPC, and the CYPA isoforms, hydroxyestradiol O, also known as estriol, is produced in abundance during pregnancy. O can be dehydrogenate to hydroxyestrone O, a metabolite that has been shown to bind covalently to the estrogen receptor via Schiff base formation. This covalent linkage occurs between the steroid carbonyl and the amino group of lysine. In theory, O could also bind DNA, although this has not been observed. O is a potent ER agonist, capable of levels of cellular proliferation stimulation that near those obtained with E. Though studies in hamster kidney tumor models showed weak carcinogenicity, the carcinogenic potential of O in humans remains unknown. As we move forward, let's uncover the untold stories and fascinating intricacies of other hydroxylations. The function of the remainder of the hydroxylated E metabolites. and O remain to be elucidated. Some of these metabolites, such as O, are excreted in relatively large amounts in pregnant women, possibly serving as an indicator of good fetal health. I hope you learned something new today. Let me know what you found most interesting in the comments below.